Anyone who has ever developed a urinary tract infection (UTI) knows that it can be painful, pesky, and persistent. UTIs have a high recurrence rate and primarily afflict women. As many as 50% of women will experience at least one UTI during their lifetime. However, what if patients could take a vaccine that would prevent future UTIs? In a study in the journal Proceedings of the National Academy of Sciences, Duke researchers describe a new vaccination strategy that they think could reprogram the body to fight off the bacteria that cause urinary tract infections successfully. Although several vaccines against UTIs have been investigated in clinical trials, they have so far had limited success. There are currently no effective UTI vaccines available for use in the US in spite of the high prevalence of bladder infections. This study describes the potential for a highly effective bladder vaccine that can not only eradicate residual bladder bacteria but also prevent future infections. The strategy, which the team showed to be effective in mouse models, involves reprogramming an inadequate immune response that the team identified last year. They observed that when mouse bladders get infected with E. coli bacteria, the immune system dispatches repair cells to heal the damaged tissue while launching very few warrior cells to fight off the attacker. This causes bacteria to never fully clear, living on in the bladder to attack again. The new vaccine strategy attempts to teach the bladder to more effectively fight off the attacking bacteria. By administering the vaccine directly into the bladder where the residual bacteria harbor, the highly effective vaccine antigen, in combination with an adjuvant known to boost the recruitment of bacterial clearing cells, performed better than traditional intramuscular vaccination. The researchers reported that bladder immunized mice effectively fought off infecting E. coli and eliminated all residual bladder bacteria, suggesting the site of administration could be an important consideration in determining the effectiveness of a vaccine. The researchers say that undertaking clinical studies to validate these findings could be done relatively quickly, since the individual components of the vaccine have previously been shown to be safe for human use.